Okay. So, I'm Shrija Ghosh. I'm the PMRF at IIT Kanpur, and I'll be taking the problem solving sessions for the NPTEL course Biochemistry. So, today is the third problem solving session, and I'll be helping you guys to solve the assignments for the week 3, that is for protein structures. So, we'll solve different questions related to the topic, and at any point feel free to ask me any questions. uh, if you guys have any doubt or at any point you don't understand anything, you can stop me and ask. You can either unmute and ask me the question, or you can write in the chat box. Okay, so I hope my screen is visible to everyone. If one of you can respond. Am I audible? Hello? Okay, I'm hoping the screen is visible now. Okay, so we'll start the class. So I'll be discussing certain questions related to your assignment only, and if you have any doubt in the questions, you can ask. Okay, so starting with the first question, question number 1: which of the following represents the two-dimensional structure of the protein? Option A quaternary, option B tertiary, option C is secondary, and option D is primary. So which of the following represents the two-dimensional structure of the protein? Does anyone know the answer? Anybody wants to answer? Which of the following could represent the two-dimensional structure of the protein? Anyone? So the answer is option D, primary. So when you have so these are basically the stages of protein folding. So you have the primary structure, which gives the two-dimensional structure. Then you have the secondary structure, the tertiary structure, and the quaternary structure. So basically your amino acid sequence, whatever the amino acid sequence are, let's say glycine, alanine. So this will form a primary sequence. This is the primary sequence. Okay, this is just the sequence of amino acids, the amino acids attached together. So this is the primary sequence. After this comes the secondary sequence, where we have the arrangement of these amino acids into either alpha sheets or alpha helix or beta sheets. Okay, either we have it in alpha helix or beta sheets. So these are the secondary structures. Then further these alpha helix and beta sheets get arranged into the three-dimensional space. So primary sequence is basically two-dimensional. Then comes the three-dimensional structures, that are secondary, tertiary, and quaternary. Okay, so secondary means how the amino acid sequence is folding into, like whether it is making a helix or a sheet or turns, loops. So those are the secondary structure. Then these helix and turns in turn join together and make a tertiary structure. Then after the tertiary structure, like different tertiary subunits can interact with each other with via non-covalent bonds to form the quaternary structure of the protein, which is the fully native folded form of the protein. Okay, so this is the native conformation. Okay, so we have 4 stages of protein folding: primary, secondary, tertiary and quaternary. Okay, so this is the basic of this is the basics of the protein structure. So whenever we are talking about protein structure, the first thing would be primary, that means the amino acid sequence. Then comes the secondary, where you have the arrangement of that amino acid into alpha helix or beta sheets or turns or loops. Then comes the tertiary, where you have the arrangement of these secondary structures in a 3-dimensional space. And then comes the quaternary structure, where further folding happens and the native conformation is achieved by a protein. Is this clear? So coming to the second question, sickle cell anemia is one of a group of inherited disorders known as sickle cell disease. It affects the shape of the red blood cells, which carry oxygen to all parts of the body. Sickle cell disease is inherited blood disorder of hemoglobin synthesis caused due to difference in the folding of a protein because of a specific genetic mutation. Identify the correct answer from the following which correctly describes this mutation. Okay, so we have a disorder known as sickle cell anemia, in which the hemoglobin synthesis is actually hampered due to a specific genetic mutation. So we have to identify that particular genetic mutation. So options are mutation in 6th position of the alpha chain, that is glutamic acid is replaced by isoleucine. Option B is mutation in the 6th position of the beta chain, where glutamic acid is replaced by isoleucine. Option C is mutation in the 6th position of the alpha chain, where glutamic acid is replaced by valine. And option D is mutation in the 6th position of the beta chain, where glutamic acid is replaced by valine. So anybody knows the answer for this? Anyone wants to answer? Come on guys, respond. Otherwise it won't be interactive. It won't be an interactive session, right? You have to ask me questions, only then you will be able to understand what is happening. Okay, so the correct answer for this question is option D, that is mutation in the 6th position of the beta chain, where the glutamic acid is replaced by valine. So what happens in sickle cell anemia? You see these rounded red blood cells. So these are the normal red blood cells or the normal erythrocytes, which are spherical in shape. Okay, but while synthesis of the hemoglobin, the gene in the DNA sequence, there is one base pair change that leads to the mutation, that leads to a faulty RNA synthesis and the protein synthesis. So I have told you about the central dogma in the first session, where we have the DNA or the gene sequence from which we get an RNA sequence. So DNA is transcribed into RNA. The process is called transcription, and we get the mRNA. From that mRNA we get protein by another process called translation. Okay, so this is basically the central dogma of the cell. So DNA to RNA to protein. So we have the DNA sequence, and in the DNA sequence, in the beta chain gene, so hemoglobin is composed of 4 subunits, 2 alpha, 2 beta. Okay. So in the beta chain, at the 6th position, the amino acid residue is replaced from glutamic acid to valine. Okay. Why this is happening? Because there's one base change in the DNA sequence. So if you see, instead of T, we have a mutation, and the base A is incorporated in the DNA. So in the normal gene, we have the code, we have the base as T, thymine, whereas in the sickle cell or the disease gene, we have the T replaced by an A, because of which what happens, now this CTC will transcribe into a codon called GAG, whereas CAC will transcribe into a codon called GUG. Now GAG codes for glutamic acid, whereas GUG codes for valine. Now what happens, in the normal protein we had glutamic acid, but in the diseased cell we have valine. Now if you remember the properties of all the amino acids we discussed in the previous sessions, you will remember that glutamic acid is charged amino acid, right? So it is hydrophilic in nature. It can easily interact with the outer atmosphere, outer hydrophilic surrounding. But if you remember, valine is a hydrophobic amino acid. So what will happen, since valine is a hydrophobic amino acid, upon deoxygenation, that valine residue, it causes the molecules to polymerize. So what happens, we have well rounded RBCs when glutamic acid is present. When the glutamic acid is replaced by valine, what is happening, a hydrophobic residue is there, so it is kind of basically the hydrophobic residue wants to go inside, and it is kind of polymerizing the whole structure, and it is making it stiff. Okay. In presence of glutamic acid, it was fuller in nature, but in presence of valine, what is happening, it is causing the stiffening of the RBCs and sickling of the RBCs. So when we called it sickle cell, it is shaped like a sickle. Okay. It is shaped like a sickle. Instead of being round, it is shaped like a sickle. So in the beta chain, so I told you hemoglobin has 4 subunits, 2 alpha and 2 beta. So 1 point mutation, 1 base change can lead to a complete disorder, which leads to sickle cell anemia. Okay. So this is the hemoglobin structure, the cartoon form of the hemoglobin structure, where we have 2 alpha subunits and the 2 beta subunits. Now in the healthy individual, the beta subunits have glutamic acid, whereas the diseased individual, the beta subunit has the valine instead of glutamic acid, which leads to the stiffening of the RBCs. Okay. So the mutation is in the 6th position of the beta chain, where the glutamic acid or glutamate is replaced by valine. Okay. I hope that's clear. If you guys have any doubt, you can ask me. Okay. Okay, moving on to the next question. Question number 3. Determining the precise arrangement of the atoms within a large protein is possible only through the use of various microscopic and spectroscopic technique. Which is or are the methods mentioned below used for structural determination of protein? So which of the following methods are used for structural determination of protein? First method is mentioned as electron microscopy, second is NMR spectroscopy, third is magnetic resonance imaging, and fourth is X-ray crystallography. So which of the following methods are used for structural determination of protein? Anybody wants to answer? Which of these four methods can be used for determining a protein structure? So the correct answer for this question is option B, NMR spectroscopy and X-ray crystallography. So till date, these two techniques have been widely used for determining the structure of protein. So especially X-ray crystallography. So most of the protein structures that are there in the protein data bank have been determined using X-ray crystallography. And in this technique, we need to develop crystals of the protein molecule. So if we have a pure protein solution, we can crystallize the proteins, and each crystal can be then used to solve the structure of that particular protein using X-rays. Okay, so it's a X-ray diffraction technique which is used for determining the structure of protein. In NMR spectroscopy, it is basically nuclear magnetic resonance, where we check the spatial arrangements of atoms in a 3D space. Okay. So both NMR spectroscopy and X-ray crystallography can give us a 3D structure of protein. What do you mean by 3D structure of protein? Basically, if I have a protein of 100 residues, I will know exactly where each residue is placed in a 3D space. Okay. So if I have 100 residues, I will know the XYZ coordinates of each residues, and thus I can tell that okay, my protein looks like this particular shape. Okay, so whether it is a globular protein or a membrane protein, how will it look, how many alpha helices and beta sheets are there, how are those alpha helices and beta sheets are arranged against each other, all those information can be achieved by these two techniques. Okay, NMR spectroscopy and X-ray crystallography. Fine. Okay, so moving on to the next question, question number 4. Consider a polypeptide of 100 residues which will have 99 peptide bonds and therefore 198 different phi and psi bond angles. If each of these bond angles can be in one of 3 stable conformations, protein may misfold into a maximum of 3 raised to the power of 198 different conformations. Therefore, if a protein were to attain its correctly folded configuration by sequentially sampling all the possible conformations, it would require a time longer than the age of the universe to arrive at its correct native conformation. This thought experiment, constituting a theory of protein refolding, is known as. So basically, if I have a protein of 100 residues, so 100 residue protein will have 99 peptide bonds. Okay, and let's say each residue can have 3 different conformations. Okay, each bond angle can have 3 different concentration. So we will have, so each bond has 3 angles, basically phi and psi angle, sorry, 2 angles, basically phi and psi angles. So we will have, if I have 99 peptide bonds, I will have 198 different phi psi bond angles, and all these bond angles, let's say, can have 3 stable conformation. So one can have 3, 198 will have 3 raised to the power of 198, like 3 into 3 into 3 into 3 up to 198 times, right? Now if a protein, if let's say I give you a primary sequence of protein, the protein will exactly know how to refold in its native conformation. It will never happen that the protein will fold into a conformation and see whether it is stable or not, and then again unfold and then again refold into the second conformation. If that was the case, the protein would have taken a time longer than the age of universe to fold, because if I have a protein of 100 residues, it will take, and if it has to if each bond angle has to undergo 3 different conformations to see which one is the most stable one, it will have to go through all the 3 raised to the power of 198 different conformations. Okay, but that doesn't happen with the protein, right? So this thought experiment was actually suggested by a renowned scientist, and this is this whole experiment is called Levinthal's paradox. Okay, option D, Levinthal's paradox. So it's a paradoxical statement because it actually doesn't happen like that, right? When a protein is folding, when we have the primary sequence of the protein already knows that it will fold in one particular stable conformation. It will never happen that it will go through all the 3 raised to the power of 198 possible conformations and then choose one. Okay, this is the one I want to be. That doesn't happen. So it's a paradoxical statement. So we see in theory that this could have happened, but this does not happen. So this is called a this whole experiment was designed by Leventhal, and it is called Levinthal's paradox. Okay, this experiment is known as Levinthal's paradox. Okay, so moving to the next question, question number 5. A homopolymer has only one type of building block called a monomer repeated n number of times. A heteropolymer has more than one type of monomer. So proteins are heteropolymers made of amino acids, while a nucleic acid like DNA or RNA is made of only 4 types of nucleotide monomers. Naturally occurring proteins in living system are made of dash. So option A is 20 types of monomer, option B is 25 types of monomer, option C is 40 types of monomer, and option D is only one type of monomer. So what do you mean by monomer? If we have all the units, so if we have repeating blocks of units, so if I have one type of unit and it is repeated multiple times, we will get a homopolymer. Okay, whereas heteropolymers means different types of units are joined together to make a polymer, that is heteropolymer. It has more than one type of monomer. So what they are saying, nucleic acid is made of 4 different types of monomers. So we have A, T, G, C, 4 bases basically, and they make 4. So A is one type of monomer, B is T is second type of monomer, and so on. These 4 monomers join together in random order to make a heteropolymer, which is the nucleic acid. Similarly, proteins are also heteropolymers made of amino acids. So basically, how many amino acids we have in nature will be the answer to how many monomers are required to make a naturally occurring protein in a living system. So naturally occurring proteins are made of naturally occurring amino acids. And how many naturally occurring amino acids do we have in nature? We have 20 types of amino acids, right? So we have 20 amino acids. So that means a naturally occurring protein in a living system are made of 20 types of monomers. It could be any of those 20 amino acids. Each residue could be of any of those 20 amino acids. So a protein is made of 20 types of monomers. So the correct answer is A. Is that clear? Moving to the next question. For a protein with 100 amino acids, how many possible sequences are there? For a protein with 100 amino acids, how many possible sequences are there? So how do we solve this question? If I have a protein of 100 amino acids, so one uh, so each residue can have 20 possibilities, right? How many amino acids do we have in nature? We have 20 amino acids. So if I have 1, 2, 3, 4, 5, 6, like that up to 100. Okay, this is the amino acid sequence. So each residue could have 20 possibilities. So first residue could have 20 possibilities, then second residue can also have 20 possibilities, then third residue can also have 20 possibilities, and so on. So we multiply all of these. So this will be the possible number of sequences. So out of 20 amino acid, one of the amino acids will be the first residue. Then out of 20 amino acids, again one of the amino acid would be the second residue. Then out of the 20 amino acids present in nature, we'll have the third residue, and so on. So total how many possible conformations or possible sequences will be there? There will be 20 into 20 into 20 up to 100 times. So 20 raised to the power of 100. So the correct answer is option D, 20 raised to the power of 100. So basically it is a general rule where if we have n residues, like we had 100 residues in this case, if we have n number of residues, the number of possible combinations would be 20 raised to the power of n. Why 20? Because there are 20 different types of amino acids that are present in nature. Okay, so in this case we had 100 residues, so 20 raised to the power of 100. If I had only 3 residues and ask you how many possible conformations could be there, so it's basically 3 residues, right? So 1, 2 and 3. So we'll have 20, like any of the 20 amino acids could be this residue, then any of the 20 amino acids could be this residue, and then any of the 20 amino acids could be this residue. So you have 20 multiplied by 20 multiplied by 20, basically 20 raised to the power of 3. So what is the general rule saying? It is saying 20 raised to the power of n, where n is the number of residues. So here the n was 3. So the answer is 20 raised to the power of 3, which is basically 8000, right? So this is the general rule. So you have to remember number of residues, whatever the number of residues is, the possible combination would be 20 raised to the power of n. Okay, this is clear. Moving to the next question, question number 7. Protein S will fold into its native conformation only when protein Q is also present in solution. However, protein Q can fold into its native conformation without protein S. So protein Q may therefore function as a dash for protein S. Now protein S can only fold in presence of protein Q, but protein Q can independently fold into its native conformation. It does not require protein S. So what is protein Q for protein S? Option A is substrate, option B is molecular chaperone, option C is protein precursor, and option D is cofactor. Anybody wants to answer? The correct answer is option B, molecular chaperone. So what is molecular chaperone? Basically something that can chaperone the other molecule. So if I have one molecule and I need another molecule to fold this particular first molecule, so the second molecule will act as a chaperone protein. Okay, so here in this case what is happening, protein S is unable to fold into its native conformation without protein Q. So it needs protein Q for folding into its native conformation. So protein Q is a molecular chaperone for protein S. Okay, protein Q is helping protein S to fold into its native conformation. But these chaperone proteins, they don't require any external help. They can fold into its native conformation without the help of the other proteins. Okay, so we have protein S that requires help, and protein Q, that is the molecular chaperone providing the help. Okay, this is clear. So molecular chaperone is basically a molecule that is helping another molecule to fold into its native conformation. Okay, it's like a helper molecule. This is clear. Any doubt till now? If you guys have any doubt, you can either ask in the chat box or unmute and ask. I hope it's clear. Okay. Moving to question number 8. Which of the following does not affect the stability of an alpha helix? Which of the following options does not affect the stability of an alpha helix? Okay, so does not affect. Okay, so you have to pay attention. For option A is electrostatic repulsion, option B is bulkiness, option C is interaction between R groups spaced three residues apart, and option D is occurrence of a proline and a glycine residues. So which of the following does not affect the stability? So electrostatic repulsion, we can say that it does not affect the stability. Just a second. Okay, there's a mistake in this question. Basically the question will be which of the following affects the stability of an alpha helix. Okay, which of the following statement affects the stability of an alpha helix? Okay, yeah, so we have an answer, option D. So occurrence of proline and glycine residues. Yes, you are right. Can you explain why? Dhanashri. It's okay. So when we have an alpha helix, in the previous class also I have told you that alpha helix forms hydrogen bonding and stabilizes its structure, right? So in these hydrogen bonding, if you see, the hydrogen bonding is happening between the amino and the carboxylic group of different residues. So in case of proline, it's very easy to understand that proline's amino group is not free. Okay, if you remember the structure of proline, the nitrogen is involved in the bond formation with the R group. So it is not a free amino group, right? And if the nitrogen is not free, it can't make hydrogen bonds, and thus it acts as an helix breaker. On the other hand, glycine has an R group, but the R group is hydrogen, so it is very small molecule. So ultimately what happens, the glycine molecule becomes very small, and becomes very flexible. It's a very small molecule, so it's a very flexible molecule. It can move in any direction at any time. So due to this conformational flexibility, it makes the helix unstable. It makes the helix unstable and leads to the breakage of helix. So glycine and proline are actually called helix breakers. Okay, Do you understand the reason? So proline is basically unable to form the hydrogen bonds, whereas glycine is conformationally very flexible, which is leading to the breaking of the helix, alpha helix. Okay. Okay. So moving to question number 9. Which given are the steps describing the hydrophobic collapse model of protein folding process. Given are the steps describing the hydrophobic collapse model of the protein folding process. Arrange them in the order in which the protein folding occurs. First is interaction of the polar surface with solvent. B is formation of alpha helices and beta sheets. 3 is formation of interconnectors and like loops and turns. And D is formation of hydrophobic core. So what is the hydrophobic collapse model? What would be the order in which the protein folding will occur in hydrophobic collapse model? Anybody wants to answer? So in a hydrophobic collapse model, by the name you can see the hydrophobic residues which makes the core of the protein, right? I have told you in the previous sessions also, if it's a globular molecule, which is there in the cytoplasm or in the aqueous environment, basically what will happen, those proteins will have hydrophobic and hydrophilic residues. The hydrophobic residues will be buried deep inside in the core of the protein, whereas the hydrophilic residues will make a outer shell of the protein. Okay. So by the name you can see hydrophobic collapse model means the hydrophobic core will form first. Okay. All the hydrophobic residues will collapse together and form the core, and then the other amino acids will form its own secondary structures, secondary and tertiary structure around it. So first step would be formation of hydrophobic core. So fourth is there in option number C and D. Then the next thing will be formation of alpha helices and beta sheets, because you can't have the next step as interaction of polar surface with solvent, because that is the outermost shell of that protein, right? So the outermost thing will happen at the end. So first step that is given, interaction of polar surface with solvent, that will happen at the last. So if you see option number C, formation of hydrophobic core, that is the first step, that is correct. Second is formation of alpha helices and beta sheets, that is also correct. Then goes formation of the interconnectors like loops and turns. So if my protein has made alpha helices and beta sheets, they have to get attached to each other by certain loops and turns. Okay, those loops and turns will form, and then at the end interaction of the polar surface with water. So the outer shell that has all the polar amino acids, they will interact with the solvent outside the protein's environment. Okay. So in the hydrophobic collapse model, basically the hydrophobic residues come together and collapse for the formation of the hydrophobic core, that is the core of the protein that is buried deep inside. Okay. And then the secondary structures will form, and then the interaction with the outer surface will happen. This is clear. So the correct answer is option number C. Right? Any doubt in this question? Okay. So moving to the next question. Just a second. So moving to the next question, question number 10th. Albumin is a large protein which circulates freely in the human plasma. Another type of protein called aquaporins acts as a channel for water to enter and exit a cell, and they are located within the lipid bilayer of a cell's plasma membrane. Based on their physiological location, how would the tertiary structure of these two proteins most likely compare? So, we have albumin, which is a large protein circulating freely in the human plasma. That means it's a protein that is living in a aqueous environment, whereas another type of protein called aquaporins, they act as channels for water to enter and exit the cell. So, they are channel proteins and they are located within the lipid bilayer of the cell's plasma membrane. So, aquaporins are membrane proteins and they are channel proteins. Okay, so they are forming channels in the plasma membrane. Okay, so let's see the options. Option A is albumin has a more non-polar core and with a non-polar outer layer, while aquaporins have more polar core with a polar outer layer. Option B is albumin has more non-polar core with a polar outer layer, while aquaporins have a more polar core with a non-polar outer layer. Option C is albumin has a more polar core with a polar outer layer, while aquaporins have a more polar core with a polar outer layer. Option D is albumin has a more polar core with a polar outer layer, while aquaporins have a more polar non-polar core with a non-polar outer layer. Okay. So, now let's see what would be the correct answer. Anybody wants to answer? What could be the correct answer for this question? So, if you see, albumin is freely floating in the plasma, human plasma. That means albumin is in aqueous environment. Okay, water molecules are surrounding this protein. So, what should be there in the surface of the albumin protein? Whether the surface should be polar or non-polar. Albumin's outer layer should be polar or non-polar? Tell me. Anybody? Albumin's outer layer should be polar or non-polar if it is in contact with aqueous environment. If it is in contact with aqueous environment, the outer layer should be polar, right? And the inner layer should be non-polar because it is away from the aqueous environment. So, albumin should have a non-polar core and a polar outer layer. This is fine. Now, let's look at aquaporins. Now, I told you it's a membrane protein. So, membrane means lipid bilayer. So, we have lipid bilayer where we have the polar head groups and the non-polar tails. So, these are the polar head groups and these are the non-polar tails. Okay, and this aquaporin is basically forming a channel between these lipid bilayers. So, water molecule can enter these channels. That means the inner layer of the aquaporin will have a polar core. Basically, the core of the aquaporin should have polar amino acids. Why? Because the core is actually forming the channel, letting the water molecules to enter and exit the cell. So, if we have a channel protein, it is basically forming a cylinder between the lipid bilayers, right? So, the inner part of the cylinder should be lined with polar residues so that they can interact with the water molecule and let the water molecule pass through it. Whereas the outer surface is in contact with the membrane, and I told you the membrane is made of non-polar tails. So, outer surface is in contact with non-polar environment. So the outer surface will be lined with non-polar amino acids. So albumin has a more non-polar core and polar outer layer, whereas aquaporins have a more polar core with a non-polar outer layer. Okay, this is clear why one is having non-polar core and why the other one is having a polar core. Aquaporins are having polar core because they have to let the polar molecules pass through it, while albumin has a polar outer layer because it has to interact with the outer surrounding that is aqueous in nature. Okay, so this is clear. So the correct answer is option B. Moving to the next question, question number 11. Which of the following statement is or are incorrect about hemoglobin? First statement is hemoglobin demonstrates higher oxygen carrying capacity compared to myoglobin. Second is there is a covalent bonding between the 4 subunits of hemoglobin. Third is deoxygenation, that is the loss of first oxygen molecule from oxygenated hemoglobin promotes the dissociation of oxygen from the other subunits. Okay, so which of the following statement is incorrect about hemoglobin? So first statement we see hemoglobin demonstrates higher oxygen carrying capacity compared to myoglobin. This is a correct statement. Okay. Second statement, if you see, there is covalent bonding between the 4 subunits of hemoglobin. What is covalent bonding? Covalent bonding, whether it's a strong bond or weak bond. Anybody knows? Whether a covalent bond is a strong bond or a weak bond. Anybody? Covalent bond is a strong bond or a weak bond? Okay, Shobha says it's a weak bond. Okay, but in reality it's a strong bond. It's one of the strongest bonds that are there in nature. So if the subunits will be attached by covalent bonding, it would be very strong, and there will be no dissociation or association or basically reforming the rearrangement of the protein subunits. Okay, so generally or most of the time, whenever a protein is folding and multiple subunits are interacting with each other, there is always non-covalent bonds between the subunits, like hydrogen bonding or van der Waals interaction. So if I have 4 subunits, so one subunit means I have amino acid sequence that has arranged in certain order. Okay, I had an amino acid sequence that has arranged in certain order and formed one subunit of protein. Similarly, I have 4 more subunits of proteins. Now these are independent units, they don't have any bond formation between them, but they have non-covalent bonds like hydrogen bonding or van der Waals interaction or ionic bonding that actually stabilizes the subunits together. Okay, so imagine like you have 4 pieces of thread. Okay, like you have 4 pieces of thread, and now those 4 pieces of thread, you make a ball out of all those 4 pieces of thread, and now all those balls you take it and take it together to make one protein unit. So each small thread balls will be basically one subunit. Okay, and those subunits are not attached together, like the thread is attached together, right? But the subunits are not attached together by any form of covalent bond. They are held together by non-covalent forces. Okay, so that is why that is the incorrect statement. So the answer is option A. There is a covalent bond between 4 subunits of hemoglobin. That is an incorrect statement, because between the subunits of any protein there is generally non-covalent bonding present. Okay. Okay. So moving to the next question, question number 12. Any doubt in the previous question or any of the questions? If you have any doubt, you can ask. You can either write it in the chat box or you can unmute and ask. Okay, at any point if you have any question, just unmute and you can ask me. Okay. So moving to the next question, question number 12. Which of the following energy term contributes to the non-covalent component of the energy in a protein structure? So in a protein structure, I told you right now we have covalent bonds and non-covalent bonds. So all the bonds, whether covalent or non-covalent, they will lead to some form of energy. Okay. So which of the following energy terms contributes to the non-covalent component of the energy? Okay. So if you see, we have 4 options here: stretching energy, bending energy, torsional energy and Coulomb energy. So if I have covalent bond and non-covalent bond. Okay. So covalent bond basically means like the bond between carbon and nitrogen or carbon and carbon. Those kind of bonds are covalent bond. Now if you have any bond between two atoms, okay, the bond will be basically like a spring. Okay, sorry. So bond will be like a spring. So let's say I have two atoms held together by a covalent bond. That bond can actually stretch or retract. So that stretching frequency will lead to a energy called stretching energy. So stretching energy is a energy term for covalent bond. Okay. Similarly, if I have a bond between two atoms, it can bend. Okay. So let's say this pen is the bond between two atoms. Imagine two atoms attached via this pen. Okay. So this pen, and imagine it as a flexible pen. Imagine it as a spring. Okay. So it can stretch. So it will have a stretching frequency. It can bend. So it will have a bending frequency also. Okay. So your spring can bend. Okay, you can bend your spring, you can stretch your spring. So all the stretching energy, bending energy, these are covalent bond energies. Torsional energy, what do you mean by torsional energy? It means the energy while rotation of anything, when where there is a torque. Okay, so if I have two bonds together, two planes together, now if I have three atoms, now if I have three atoms, let's say 1, 2, 3 and 4. If I have 4 atoms, so there will be two planes in this 4 atom. 1, 2, 3 will make one plane, and 2, 3, 4 will make second plane, and there will be a angle between these two planes. What is that angle called? I have told you in the previous class that angle is called dihedral angle, and those angles can actually give a rotational energy, which is called the torsional energy. So if I have two planes joined together like this, it can actually, one plane can rotate with respect to another plane in different direction. Okay, so the bond can basically rotate around each other. Okay, and that rotational frequency is called the torsional energy. Okay, so that is also involved in the covalent bond energy. The last option, Coulomb energy, is actually, Coulomb is a unit of charge. So it's actually energy associated with the ionic bonds. Okay, so ionic bond, it's a non-covalent bond basically. So that Coulomb energy is the energy term for non-covalent bond. So the correct answer is option D, Coulomb energy, that contributes to the non-covalent component of energy in the protein structure. So if you see, I explained bending energy, torsional energy, the rotation, and the stretching energy. Okay, these are all covalent bond energy. Non-covalent bond energy is basically the bond between the two atoms that are spatially apart, and there's a bond between the hydrogen and an electronegative atom, so that is a hydrogen bond. Or there is a van der Waals interaction between two atoms, or there is an ionic bond where there is a positive molecule and a negative molecule, and they are attracted towards each other due to presence of the charge. This ionic bond energy is basically the Coulomb energy. Okay. Other than Coulomb energy, other non-covalent energy terms are hydrogen bond energy or van der Waals interaction. Okay. This is clear? Okay. Any doubt? Okay, so moving on to the next question, question number 13. In the given hydropathy plot for a certain protein, how many distinct segments of amino acid will form hydrophobic parts of the protein? So I have discussed about hydropathy index and Kite-Doolittle plot in the previous session also, so you should be able to answer this. So I have told you, in a hydropathy plot, hydrophobic residues have a positive value and the hydrophilic residues have negative values. Okay, so in this plot you can see in total how many sections? We can see 1, 2, 3, 4, 5, 6 and 7 sections are there. Okay, total 7 sections are there. Out of 7 sections, how many sections are hydrophobic and how many sections are hydrophilic? Tell me, how many sections are hydrophobic in this plot? I have told you, hydrophobic residues show a positive value in the hydropathy plot. Yes, Shobha has answered 4. Very good. So 4 sections, we can see 4 sections are above the axis. So 1, 2, 3 and 4. So these 4 sections have a positive hydropathy index. Thus, those 4 sections represent the hydrophobic parts of the protein. Is that clear? So I've shown you this in the previous class also. We have different hydropathy index for different amino acids. So all the non-polar amino acids are basically hydrophobic amino acids and they have positive values or slightly negative values only. For glycine and proline we have slightly negative values because they are kind of an exceptional amino acids. Glycine, being a very small molecule, cannot determine the hydrophobicity of a molecule, whereas proline, even after being a non-polar amino acid, the amino group is not freely available, and thus sometimes it leads to a negative hydropathy index. Okay, other than that, if you see all the other polar groups, most of them have negative negative hydropathy index because they are, the charged groups are highly hydrophilic and they show a negative hydropathy index. Okay, so you can see the 4 sections that are on the top of, like, that are above the axis that have the positive hydropathy index are showing the hydrophobic parts of the protein. The 3 parts that are below the axis, they are showing the hydrophilic parts of the protein. Okay, hydropathy plot is actually a very important method to determine how many regions of the protein are hydrophobic and how many regions are hydrophilic. After doing the hydropathy plot analysis, you can actually determine, okay, these many sections are hydrophobic, so they are buried inside the protein. If it's a globular protein, it is in the core of the protein, or if it's a membrane protein, it is in the outer layer of the protein. Okay, so like those kind of informations can be gained by such plots. Okay, any doubt? I hope this is clear. Okay, so moving to the next question. Question number 14th. Predict the structure of predict the secondary structure of the given peptide. The peptide is given chemist, CHEMIST. I have given you the table that is showing the Chow-Fasman parameters. What are these parameters? These are basically the probability of an amino acid to become an alpha helix, a beta sheet, or a turn. Okay. So if you see for alanine, the probability of alpha helix is given, the probability of it becoming it getting incorporated into a beta sheet is given, and probability of alanine uh, getting incorporated into a turn is given. So we have to predict the secondary structure of this peptide. How can we solve this? So what do we have to do? We will have to first write the values, the PA, PB and P-turn values for each of these amino acids. So what is C? C is cysteine. So I can see the probability of it becoming alpha helix is less than 100, but for it to become beta sheet or turn is more than 100. Okay, so like that I will see for each and every amino acid. So what is the rule? We have to see 6 contiguous amino acids, and out of those 6, if 4 amino acids have a high a probability value or the PA value more than 100, we can say that that stretch of amino acids, like that particular stretch of peptide, will form alpha helix. Okay, if same thing is happening, we will do the same thing for beta sheet. We will take 5 contiguous amino acids, and 3 out of 5, if they are showing probability of B, that is PB, as more than 100, we will say that the sequence will form beta sheet. Now if you see, now we will see first we will see the analysis. So we have the amino acid chemist here. Okay, so C is cysteine. So I have written the values from that table only, so you can also see the values. So for cysteine, probability of A is 70, probability of B is 119. So I have written here probability of alpha helix is 70. The propensity is for beta sheet is 119. Then H is histidine. Let's see, for histidine the value is 100 for PA and PB is 87. So I have written 187. Similarly, E is glutamic acid. So I have written the values of glutamic acid. Then M is methionine. So methionine, I have written the values for methionine there. Then I is isoleucine. Isoleucine, the value is PA is 108 and PB is 160. Okay, so probability of it becoming or getting incorporated into a beta sheet is higher than alpha helix. Okay, then S is serine. Serine is 77 and 75. So probability of it becoming alpha helix or beta sheet both are pretty less. But if you see the probability of it becoming a turn is more. Then last is T. T is threonine. Threonine is probability of alpha helix is 83, but beta turn is beta sheet is 119. So let's see, I have written the values here. Now what does the rule say? We have to take 6 contiguous amino acids and see out of those 6 if 4 are more than 100. So I'll take 1, 2, 3, 4, 5 and 6. Sorry. So from C to S, if you see 1, 2, 3, 4, 5, 6. So out of these 6, if I see more than 100. So 1, 2, 3 and 4. So 4 amino acids out of 6 contiguous amino acids have more than 100 p value for alpha helix. That means this particular stretch of peptide might form a alpha helix. Now let's see for beta tone. Let's see for beta tone also. Beta sheet also. I'm sorry. Let's see for beta sheet. 1, 2, 3, 4, 5. So out of 5, 3 are more than 100. So in this case, there's an overlap. What it says about the overlap? If alpha and a beta region overlaps, the overlapping is predicted according to summation of PA and PB. Okay. So we will have to add the PA values together and the PB values together, and then we will have to see whether whether the PA value is higher or the PE value is higher, whether the sum of PA value is higher than the PB value. Okay, is that clear? So if I see the values here for 6 contiguous amino acids, out of those 4 are above 100. If I add all these values together and if I add all these values together, you will see P alpha is obviously higher than the P beta sheet. That means this particular stretch of peptide has more probability of becoming into an alpha helix than into a beta sheet. Is this clear? This is how, if you have a peptide sequence given and the Chow-Fasman parameters given to you, you will have to see for each of the amino acid what is the PA or PB values, and then you will have to see whether those PA or PB values are more than 100 or less than 100. If 4 out of the 6 contiguous amino acids have a more than 100 value for PA, that means the peptide might form a alpha helix. If it has 3 out of 5 amino acid residues with PB higher than 100, it can form a beta sheet. If both the cases are there, then you do the summation. So summation of PA and summation of PB. If the summation of PA is higher than summation of PB, it will form an alpha helix. If the case is opposite, that is summation of PB is higher than PA, then the chances of it becoming a beta sheet is higher. Is that clear? Any doubt in this question? If you have any doubt, you can ask me. I hope this part is clear. Does anyone have any doubt? Okay. So moving to the next question, question number 15. What would be the pI of aspartic acid if pKa1 is equal to 2.1, pKa2 is equal to 3.9 and pKa3 is equal to 9.8? Okay. So I have the structure of aspartic acid given at different pK. What is the charge, that is also given. So I will have to calculate for the pI of this aspartic acid. Okay, and pK values are given at every state. So anyone knows how to calculate? What will be the answer for this? What is pI? Anybody, what is pI? So pi is basically the point where the protein, the pH where the protein or the peptide or the amino acid has the net charge as 0. Okay. So if I plot a pH titration curve for amino acid, so if I keep on increasing a pH, so this is the pH scale, that is OH negative concentration. Sorry, this is the pH scale and that is the OH negative concentration. So if I keep on adding base, after a certain point protons will dissociate, and when there is no more proton left, there will be a sudden increase in the pH till the point the next pK value is reached. Once the next pK value is reached, again the protons will be released to neutralize the pH. Once all the protons are consumed, there will be a sudden increase in the pH again until the next pK value is reached. So these are basically, this is basically the pK values at each stage. Okay. So isoelectric point, that is pi, is basically the pH at which charge is 0. The net charge is 0. Okay, net charge is 0. So if you see here, just a second. Okay. So if you see here, pi is basically the pH at which net charge is 0. So if you see here, I have an amino acid that is fully protonated. The amino group is protonated, the carboxylic group is protonated, and the side chain is protonated. At a very low pH, everything will be protonated. Now if I keep on adding base to this, what will happen? It will start dissociating one of the protons. So first the carboxylic proton will be dissociated. So it will keep on dissociating that proton in this region and neutralizing the pH. Once all the protons are released, it reaches this stage. Okay. At pK1, it reaches this stage. Once all the protons are released, there is a sudden increase in the pH. Okay. At pK1. Now what happens? Now this proton is not there. Now again, if I keep on adding base, it will start losing another proton from the side chain till we cross this pK value. Once this pK value is crossed, no more protons are left for this group to give, and there will be again a sudden increase in the pH. Then again the amino acid will start losing the third proton, that is with the amino group, till the third pK is crossed. Once that pK is also crossed, there will be no more proton left to release, and there will be an increase in pH and then a stable graph. Okay. So the pH will reach up to its maximum point. Okay, that's clear. So now I am saying the pi is basically the pH at when the net charge is 0. So if you see the charge in all of these states, here the charge is +1, here the charge is +1 from this, -1 from this, and 0. So +1 -1, that is 0. So net charge is 0 at this stage. So this stage is coming between pK1 and pK2. So pi is basically the average of these two pK values. Okay. So imagine the pi to be a wall. Okay. Imagine a pi to be the wall, and pH, if the pH is on the left side of the wall, everything will be protonated. Okay. If you cross this wall, if the pH crosses this wall, groups will start getting deprotonated. Okay. So when we cross pK1, first group, the pK1 value is for this group, so this group will get deprotonated. Then when we cross pK2, when we cross this wall, the second group will get deprotonated, because pK2 is basically the value for this proton. Then when we cross pK3, pK3 is basically the value for this proton. So when this proton is when this pK is crossed, this group will get deprotonated. Okay, so like that, if you now see, the net charge is 0 at this stage. This stage is coming between pK1 and pK2, between these two walls. So the Pi will be somewhere in between these two walls. So how do we find the midpoint of any 2 points? Basically taking the average of that, right? So if I have pK1, if I have the value of pK1 and pK2, what will I do? I will take the average of that. Okay, so let's see how to solve. So if I have, so the net charge I told you for this form, it is +1, because +1 from this, and then these are uncharged, these are at 0. Then you have this form that has +1 and -1, so net charge is 0. Then you have this form where +1, -1 and -1 from the side chain as well. So the net charge is +1 and -1 gets cancelled, and there is one more -1. So the net charge is -1, and here the net charge is 0, -1 and again -1. So -1, -1, -2. So the net charge is -2. So if I have to calculate the Pi, I will basically do pK1 plus pK2 divided by 2. That is the average of these two pK. So pK1 is given as 2.1. If you see in the question, pK1 is given as 2.1, pK2 is given as 3.9. So 2.1 plus 3.9 divided by 2. So 2.1 plus 3.9 is 6, and 6 divided by 2 is 3. So the correct answer is C, option C. 3 is the correct answer. 3 will be the Pi of aspartic acid. Is that clear? Is that clear to everyone? So 3 is the Pi of aspartic acid. We just have to see which form is uncharged, net charge is 0. That particular form, we will see the pK before and after that particular form, and we will take the average of those two pK values. Okay. So let's say if I had a basic amino acid. So this was aspartic acid. If I had an asparagine, okay. So let's say I have a basic amino acid. So I will have a amino group, a carboxylic group, and it's a basic amino acid. So there will be a amino group in the side chain also, right? Now if I ask you the net charge on this, the net charge will be +2, and then when deprotonation happens at first pK1, we will have the amino group will the carboxylic group will lose the proton first, then our second amino group will lose the proton first, and then the last amino group will lose the proton. So from +2 we will reach +1, then 0, and then -1. So in that case, the average will be taken between pK2 and pK3. So if you have a basic amino acid, you will have to take the average of these two pK. If you have an acidic amino acid, you will have to take the average of these two amino acids. Is that clear? So it's not always like you will have to take the average of the first 2 pK. You have to see where, at what point the net charge on an amino acid is 0. At that particular point only you will have to take the average of those 2 pK before and after that particular form. Okay. So I hope this is clear how to calculate PI. This is a very important topic, so you need to understand. If there's any form of doubt, you can always stop and ask me. Okay. This is clear to everyone? Okay, then we'll move forward. Next question is question number 16. Which of the following best represents the backbone arrangement of two peptide bonds? Okay. Which of the following best represents the backbone arrangement of two peptide bonds? So we have here in the first option we have a C alpha attached to the amino nitrogen group, then again to C alpha and then carbon. In the second option we have a C alpha attached to the nitrogen, then to carbon, then again to carbon, nitrogen and C alpha. Third option we have a carbon that is attached to nitrogen, that is attached to a C alpha, which is again attached to a C alpha and carbon and nitrogen. And option D is we have C alpha attached to a carbon, attached to a nitrogen, then again C alpha, carbon, nitrogen. Okay. Anybody wants to answer this question? How would be the peptide bond backbone, a peptide backbone will look like? We had discussed this in the previous session also. So, anybody who attended that and remembers? So, a peptide bond basically is made between two amino acids, right? So, if I have one amino acid with a nitrogen attached to a alpha carbon attached to a carboxylic carbon, then this carboxylic carbon will form the peptide bond with the next amino acids amino group. So, nitrogen, again C alpha, again the carboxylic carbon. So, this is one peptide bond. Okay. So, this is basically one peptide bond formed between two amino acids. Similarly, one more peptide bond would be looking like this. Okay. So, now let us see which of the following options are matching this. So, C alpha, after C alpha they have given N. So, after C alpha we have a C, here we have a N here. So, if it says, even if it is going backwards, so C alpha N, then comes C. So, C alpha N and then again C alpha. So, this is wrong. Okay. So, first option is wrong. We see the second option, C alpha. Okay, we have a C alpha, then N. Okay, we are, if we go backwards, then N, then again C, that is also fine, then again C. So, after C we will have again C alpha. It is going in a repetitive order, N C alpha C, then again N C alpha C. Okay. So, this is also wrong. If you see this, we have C N C alpha, this is fine, C N C alpha, then again C alpha. So, this is also not the right structure. The right structure is basically Priyanka, do you have any doubt? Do you have any problem? Priyanka? please unmute and ask whatever problem you are having.
मैम ये ट्रांसलेशन का ऑप्शन नहीं खोल रहा।
कौन सा ट्रांसलेशन?
इंग्लिश टू हिंदी।
ऑडियो ट्रांसलेशन?
यस मैम।
हाँ, जस्ट अ सेकेंड। आई डोंट थिंक ऐसा कोई ऑप्शन है, मुझे चेक करना पड़ेगा. We have been asked to take the class in English only primarily. All the lectures are also recorded in English. So,
ओके मैम।
once I'll upload the video on YouTube, you can use the caption, closed captions on YouTube. उसमें दिख जाएगा,
ओके मैम।
ठीक है, उसमें
ओके
ट्रांसलेशन दिख जाएगा, ठीक है.
थैंक यू मैम।
Yeah. Okay, so the correct answer for this question is option D. Because you see in option D we have C alpha Cn, then again C alpha Cn, so it's a alternative unit of the same sequence. So C alpha Cn, then C alpha Cn, and so on. Okay, so correct option for the best representation of a peptide backbone is option D. Moving to the next question, question number 17. For the portion of a peptide shown below, which of the following statements is true? So we have a peptide shown here. Which of the following statement is true? So X, Y, Z, we have 3 bonds labeled. So which bond is showing what angle, we have to tell. So anybody wants to answer this question? So if you see, here we have again a carboxylic carbon, the amino group of the next amino acid, the C alpha, and again the C. So the bond between the amino and the C alpha, that bond angle, that bond's rotational angle or the dihedral angle, it is called phi, which is labeled by Y. So this Y is basically nothing but phi angle. And if you see the next angle, that is the angle between the C alpha and the carboxylic carbon, that angle is called the psi angle, that is labeled by Z. So the correct answer for this question is option B, phi and psi angle, that is labeled by Y and Z. So here phi is the torsional angle around the N C alpha bond. So this is the C alpha here. So N C alpha bond, the angle the torsional angle is Y, that is torsional angle that is labeled by Y, it is the phi angle. And the torsional angle between C alpha and C, that is C alpha and the carboxylic carbon, that angle is called the psi. Okay, that angle is called the psi angle, which is labeled by Z. Okay, so the correct answer is B, Y is phi angle and Z is psi angle. Okay, this, I hope this is clear. Okay, moving on to the next question. Question number 18. An unspecified protein contains a 18 residue long alpha helix. How many full turns are there in this alpha helix? And what is the length of the alpha helix. Okay, so we have a 18 residue long alpha helix. Okay, so we have a helical structure made of 18 amino acids. Now those 18 amino acids will, how many out of those 18 amino acids, how many will make up one turn? You have to know that. Then only you can calculate how many full turns will be there in the whole alpha helix. The next question is, what is the length of the helix? So you should know how many residues make up, like there's always a, like there's a term for alpha helix. If I make a alpha helix for you, so I'm saying 18 residues are making this. So 1, 2, 3, like that we will have 18 residues making this alpha helix, right? So these residues, there is a definite term that is making one turn, like certain angstroms, 5.4 angstrom give rise to one turn, and if you see 3.6 residues actually contribute to one turn. Okay, so this is one turn. Okay, so now how do we solve this question? Anybody? So I have told you 3.6 residues are making one turn, and one turn is 5.4 angstrom long. Okay, so we have two questions here. How many full turns are there in this alpha helix, and what is the length of the alpha helix? Okay, so to answer the first question, that is how many full turns are there in the alpha helix, so I have told you in one turn there are 3.6 residues. So 3.6, so 1, 2, 3, and the fourth residue comes in the second turn. So 3.6 residues come in one turn. Okay, so how many total residues we have? We have 18 residues here. We have 18 residues given in the question. So 3.6 residues are making one turn. The next 3.6 will make second turn. So that will make 7.2. Then again 3.6 will make the third turn. Like that, when you will keep on going, so you will see how many turns will make 18 residue long alpha helix. So basically you have one turn has 3.6 residues. So one residue will have 1 by 3.6 turns, right? And 18 residues will have how many turns then? 18 by 3.6, right? So 18 by 3.6 turns. So if you divide 18 by 3.6, you will get the answer to be 5. Okay, so number of residues in one turn of alpha helix 3.6. Given number of residues is 18, so number of turns is 18 divided by 3.6. So 3.6 into 5 is 18. So number of turns, so total number of turns is 5. Now the length of the alpha helix. So I told you one turn is 5.4 angstrom. So if I have 5 turns, it will be basically 5.4 into 5. 5 times 5.4. So one turn is 5.4 angstrom. So length of the total helix, like 18 residue long helix, will have 5.4 into 5, that is 27 angstrom. So we will have 5 turns in an 18 residue long alpha helix, and the length will be 27 angstrom long. I hope this is clear. Do you understand how to calculate? Is there any question? Any doubt? Yes, Satya, you have any question? Hello? Okay. Okay, if there's no more question, I'll move to the next question. That is the last question. So, question number 19. How many hydrogen bonds between the backbone atoms are there in a 15 residue helix? So, now if you remember in the previous class also I have told you where the hydrogen bonds form in an alpha helix. In an alpha helix, we have the hydrogen bonds between which two residues? If you remember, we have discussed in an alpha helix, we have the carboxylic group of ith amino acid forming the hydrogen bond of with the amino group of I plus fourth amino acid, right? Right. So if I have 15 residue long alpha helix, so how many hydrogen bonds we will have? Yes, first and fourth. Very good, Ramya. So first and fourth, uh, first and fifth actually. First and 1 plus 4. So first and fifth will make the hydrogen bonding. Okay. It's not first and fourth, it's first and fifth. I and I plus 4. So 1 will make the bond with 5, 2 will make the bond with 6 and so on. Okay. So uh, here if you see the number of hydrogen bonds, we will have we have 15 residues. So let's say 15 residues can make up to 30 so there will be maximum two hydrogen bonding present, because if I am saying N is making hydrogen bond with C and C is making hydrogen bond with, basically NH is making hydrogen bond with C. Just a second. Yeah. So basically we have amino group that is making the hydrogen bond with the carboxylic group, and the carboxylic group is making the hydrogen bond with the amino group, right? So in an amino acid we have both amino and the carboxylic group, right? Right now we discussed the peptide backbone structure. So this will also form a hydrogen bond and this will also form a hydrogen bond, right? So one amino acid can have maximum 2 hydrogen bonds. So that's why for 15 residues we can have maximum 2 hydrogen bonds for each residue. So that will be 30 hydrogen bonds. But now if you see an alpha helix, now let me draw a 15 residue helix for you. Just a second. So if I now make 15 residues, so we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 and 15. We have 15 residue long alpha helix. Now if you see, So this is the N terminal and this is the C terminal. Okay. Now if you see, this N terminal and C terminal will be free. Okay. So the hydrogen bond will start with the carboxylic group of this. So C double bond O, carboxylic group of this. So this is the first amino acid. So 1, 2, 3, 4, 5. So this is the fifth. So if this is I, this will be I plus 4, right? 1, 1 plus 4, right? So this will make hydrogen bonds with the fifth residue. So this will make only one hydrogen bond. Similarly, the second will also make only one hydrogen bond with this. So in an alpha helix, always the terminal 4 amino acids. So 1, 2, 3 and 4. These 4 amino acids will make hydrogen bonds with only one amino acids, because they don't have any amino acids this side. So they don't have any I plus 4 or I minus 4 amino acid on this side. Whereas from the fifth residue, if you see, fifth residue, one hydrogen bond is already being made with the first amino acid. The second you will see, so this is fifth, right? So 5 plus 4 would be 9. So 6, 7, 8, 9. So fifth can also make a hydrogen bond with the 9. So from the fifth residue, you will see it is making 2 hydrogen bonds, right? So we have first 4 residues making only one hydrogen bond. Similarly, last 4 residues will also make only one hydrogen bond. So if this is there, then it will make a hydrogen bond with this. And similarly, and so on. So if you see, first 4 and last 4 amino acids will make only one hydrogen bond. So that is why in an alpha helix, 4 residues at the N terminal and C terminus can make only one peptide bond. That means, sorry, this will be one bond per residue, basically one peptide bond. So sorry, one hydrogen bond, I'm sorry. So that means out of these 30 hydrogen bonds that we had calculated, we have to remove 8 hydrogen bonds. So 30 minus 8 is 22. Now out of 22, if we are calculating, basically what we are doing, we are calculating this as 1, 2, 3 and 4. Then for fifth one, we are calculating this twice. We are calculating this also again, which we don't want to do, because we have already counted this. So we will just count this one. So if you have to count only this one, so the counting that we are doing right now is twice for these center amino acids, right? So we have to remove that. So we will basically divide it by 2, because we are doing the counting twice. We will divide it by 2. So that will give the final number of hydrogen bond to be 11. Okay. If you now manually count the hydrogen bond, you will see there are 11 hydrogen bonds. So this is the first hydrogen bond, okay, 1. Then this is the second hydrogen bond, 2. This is the third hydrogen bond, 3. This is the fourth hydrogen bond, 4th. Okay. Then fifth, this we have already counted. So this is the fifth hydrogen bond. Then for 6, this we have already counted. So we will count this one. Okay, this is the 6th hydrogen bond. Then for 7th, we will count this one. This is the 7th hydrogen bond. And for 8th, we will count this one. This is the 8th hydrogen bond. Okay. So for 8 also we have counted this. Now for 9, we have already counted this. Okay. So 9 plus 4 means 9, 10, 11, 12, 13. So 10, 11, 12, 11, 12, 13. So 9 will make this hydrogen bond. So this will be the 9th hydrogen bond. Then this will be the 10th hydrogen bond, 11th hydrogen bond, and that's it. This is the last residue. This is the 15th residue. Okay. So total we have 11 hydrogen bonds in a 15 residue. For 15 residue helix, alpha helix, we have 11 hydrogen bonds. Is this clear? I hope this is clear. So for 15 residues, we have 11 hydrogen bonds. Okay. So the calculation is simple. Basically, you will have to see how many residues are there. Multiply it with 2 first. So that many. Then you just subtract 4 from each terminal. So 4 from the N terminal and 4 from the C terminal. Once you subtract those, you will get the number of hydrogen bond, but that counting will be twice, because you are counting like the fifth amino acid or the sixth amino acid, they are making 2 hydrogen bonds. So you are basically counting it twice. So now you just divide it by 2 to get the uh, actual number of hydrogen bonds in the alpha helix. Okay. So this is clear. So the correct answer would be option A, 11. Okay. So this was the last question. Any doubt in any of the questions discussed today? So the concept of alpha helix, how the hydrogen bond is forming, and the concept of pi, how to calculate a pi, then how to make a helical wheel, that I have discussed in the previous session. Those things are very important. How to determine the how to analyze a hydropathy plot, or how to check whether a protein sequence is making a alpha helix or the beta sheet. So those are actually the things that are going to tell you about the protein sequence, like protein structure, how to determine a protein structure. So this is how you will proceed towards solving a protein structure. So these concepts are very important for this topic. Okay, so hope you will upload the, yes, we'll upload the session in the portal. So I think already the link has been uploaded in the portal, so I'll just paste my YouTube link on this, on that portal only, and you can access the video. Okay, the session has been recorded, so that will be uploaded, and I'll upload the PPT also so for your reference. Anything else? Anybody has any doubt, then you can ask me, we can have a discussion. If there is no more doubt, then we will end this session today. Thank you everyone for joining.